Okay, I think we can start the last part of our course. So now we're going to move to preclinical imaging, post-mortem, and histological validation. And the first speaker is Manisha Hargval, and she's going to talk about preclinical imaging and post-mortem diffusion imaging. Thank you.
So thank you for the introduction. Hello, everyone. So this morning, we've heard all about diffusion acquisition, modeling, and analysis. So now I'm going to slightly shift gears and move towards preclinical and post-mortem diffusion MRI. So this is the very standard Stetskil-Tanner sequence for diffusion using pulse gradients. And if you use the simplest Gaussian model of diffusion, we can fit the signal exponentially with the b-value. And from this, we can calculate the ADC, or the apparent diffusion coefficient of water. And these are some three-dimensional reconstructions of electron microscopy images that show the typical tissue microenvironments in brain tissue. So most of what we do in diffusion MRI is aimed at understanding the relation between this diffusion signal and tissue microstructure. And this is an area where preclinical imaging is important because it allows us to directly investigate this relation. And it also allows us to somewhat push the limits of diffusion MRI beyond what is typically possible in the clinic. So here I'll start with some of the technical considerations that we need to bear in mind when moving to diffusion on preclinical systems or at high fields. And then I'll introduce several applications that highlight how different diffusion methods can be used to probe brain tissue microstructure. And in the last section, I'll introduce diffusion using oscillating gradients, which is a relatively recent and interesting area, and show you how we can use it to look at microstructure in the brain. So for MRI, moving to higher field strengths is always considered desirable because we can get a gain in the signal, or the SNR. But there are also some technical aspects that we need to know, and this is especially true for diffusion. So preclinical field strengths today are anywhere between 4.7 to 21 Tesla. And as we move to higher fields, so we get stronger field inhomogeneities. Importantly, the tissue T2 and T2 star both become shorter, whereas the T1 becomes longer. And what this means for us is that clinically feasible echo times cannot generally be used because they result in signal loss. And for diffusion, this means that we also need to use shorter diffusion times. And then, of course, we get more eddy current distortions and more severe susceptibility artifacts. So all of these also limit the application of single-shot API that we heard about this morning, which could have allowed faster acquisitions. So Dr. Miller covered all about acquisition this morning, so I'm only very briefly going to touch upon a couple of additional things that we need to know for diffusion on preclinical systems. So for acquisition, we can use any of the standard sequences that we have, and this depends on your scan time, resolution, T2 decay, and noise tolerance. And then for diffusion gradients, we can use the very conventional spin echo that you've seen before, or we can use bipolar diffusion gradients that I've shown on the right. And the advantage of using the second scheme is that we can reduce the effect of eddy currents, which can be problematic at high fields. And this is simply because here we are using these positive and negative pairs of gradients, which can cancel out some of the effects of eddy currents. And the second thing I'm going to touch upon is, and what you need to know is that a major problem for using multi-echo sequences on high fields is that we get severe phase errors in the images. And this can happen due to eddy currents or physiological motion, which basically causes the phase to oscillate between even and odd echoes. And one way to fix this is to use navigator echoes. So here on the right, I've shown an example of images before and after correction. So here you can see in the image before correction, there are severe blurring artifacts next to the eyeball. And this is something that is typical of diffusion-weighted images at high fields. And then we can see that these artifacts could be effectively removed using navigator correction. And then we can get this clear and sharp image on the right. So now I'm going to show some very recent examples of how different diffusion methods can be used to probe microstructure in the postmortem brain. And this will also bring together some of the techniques that you heard about this morning and show their more practical applications for imaging the brain. So this is an example of using diffusion spectrum imaging in the macaque brainstem. And if you remember, DSI is basically a model-independent approach, and it typically requires a very large number of diffusion-weighted images to be acquired. And this involves changing both the magnitude or the B value of the gradients as well as their orientations. So in this case, 515 images were required. And here, if you look at on the left, we can see that the fibers reconstructed from DSI very nicely show these crossing fiber populations in the pons. And in comparison, if you look at DTI on the right, we can see that the tensor model of diffusion is somewhat inadequate to resolve these crossing fibers in this region. And next, this is data from a human brainstem. So here we can see the small region of low FA that is indicated by the white box. And then we can see the zoomed-in view of the reconstructed fiber orientation distributions from Hardy in this region. And now if we further zoom on a very small area, so here we can get a clear picture of the complex fiber orientations that can be resolved in the reconstructed FODFs. And so what could we do with these? So based on this, we can also do probabilistic tracking and generate a track density image. So here again, we can very clearly see the crossing fiber populations that are resolved using Hardy in the human brainstem. So these two slides basically highlight some examples where Hardy methods can provide additional information in the brain compared to a more conventional or simplistic diffusion tensor model, or DTI. So why is this useful and why bother? So diffusion contrasts in the postmortem brain can actually also help us to better understand in vivo diffusion MRI contrasts. So for example, here I've shown an in vivo image, which is acquired on a clinical scanner at 3 Tesla. And then if you look at the postmortem image of the brainstem acquired on 11.70, so here we can clearly identify the major white matter tracts in the brainstem. And also here we can differentiate these subtle differences in their orientations. For example, we can differentiate, I'm not sure if you can see this. We can differentiate the medial lemniscus and the superior cerebellar peduncle in this blue region here. Try to use this. Okay. So here we can differentiate the medial lemniscus and the superior cerebellar peduncle in this blue region over here. And the next thing to note is that this ex-vivo data is acquired using 12-shot EPI as compared to single-shot 2D EPI in the in vivo image. And this is because we are imaging at high fields and also because we are dealing with fixed tissue. So both of these actually independently cause much shorter T2s in the tissue. So this is one thing that we need to keep in mind when we are going to move to postmortem imaging. The second thing to note is the difference in B values. So this in vivo data are acquired at a B value of 1,000, whereas the ex vivo image is acquired using a B value of 4,000, which is almost 4 times. And this is because in addition to having shorter T2, fixed tissue also has much lower ADC, or apparent diffusion coefficient of water. And this is important to know because this means that we need to use much higher B values to get comparable contrasts in the postmortem brain. So here again, we can see very fine fiber orientations that are resolved in the ex vivo image. For example, these fine crossing or interleaving fibers of the pons, which are seen in red here. So this can then help us to better interpret or understand corresponding diffusion contrasts in the in vivo image, which I've shown on the left. So now moving in the other direction, diffusion contrasts in the postmortem brain can also be directly compared with histology or myelin staining to understand the microstructural basis of the diffusion contrasts. So here I've shown an example comparing myelin staining with diffusion color maps of the brainstem. So here we can identify and compare major myelinated and white matter tracts. And then this is just to give you an example of areas where diffusion can sometimes also provide additional information compared to what we can get with myelin staining. So here we can see these very fine horizontal red fibers that are emerging from the olives in the medulla. And these are less easy to differentiate using myelin staining because they basically just differ in their orientations. So now diffusion, of course, can be used to delineate white matter tracks. There's not much new in that. But can we actually use diffusion to go beyond and look at gray matter areas in the brain? So one recent area of interest is to map cortical areas in the human brain. And this is a standard or familiar map showing Brodmann areas in the human brain. And on the right is a classical drawing by Cajal that shows the layered structure and horizontal fibers in the primary visual cortex. And looking at this figure, we can also get some idea of why diffusion in gray matter areas is more complex. This is because we have less diffusion anisotropy because of cell bodies and dendrites as compared to white matter. So now I'll try and show some examples of how we could use diffusion to probe cortical microstructure in the human brain. So this is a recent study using Hardy of ex-vivo human visual cortex specimens. And this is Hardy using 60 directions. So in this figure, we can see that based on the fiber orientation distributions, the visual cortex could be divided into 4 layers. And here on the right, we can also see intracortical fibers that are reconstructed in the stria of Gennari. And then this figure also compares these fibers using polarized light imaging and myelin staining to try and understand the diffusion orientations in the cortex. So this is another example showing how we could use diffusion to actually differentiate different cortical areas within the visual cortex. So here, if we look at the color map on the left, we can see a clear layered structure in the primary visual area that is resolved on the basis of diffusion contrasts. And this is in stark contrast to what we see in the adjacent secondary visual area. So based on this, we can now actually demarcate the boundary between these two cortical areas in the visual cortex. And then again, we can go ahead and look at comparison with silver staining in the same specimen to try and understand what these diffusion contrasts mean. So here on the right, I've shown silver stain sections. So here we can see horizontal fibers in 2 particular cortical layers that are running parallel to the cortical surface in the visual cortex, or in the primary visual area to be specific. And then we can see on the right that the secondary visual area shows predominantly radial fibers. So this can then help us understand what the diffusion contrasts in the ex-vivo brain mean. And this is also an example that highlights how combining high angular and spatial resolution can be used to probe microstructure in the post-mortem brain. So these are some quantitative plots showing the cortical profiles of fractional anisotropy, which basically again show a clear difference between the 2 areas. And here, if you look at the plot on the left, which is from the primary visual area or area 17, we can see a nice laminar profile that is delineated. And we can also see low FA in particular areas of the cortex. So what about the fiber orientation distributions? So if we go ahead and look at fiber orientation distributions reconstructed from Hardy, so here the first thing that we can see is that the distributions are a little more complex than what we would expect in typical white matter fibers, for example. And here we can also see the tangential orientations that correspond to crossing fibers in the stria of Gennari that are clearly resolved. And looking at this map, you can also see the layer specificity of orientation distribution functions in the visual cortex. So then the question is, can we go ahead and use Hardy to actually differentiate cortical areas in the human brain? So this figure shows a comparison of track density images of the primary motor and visual cortices. And this is data acquired using single shell Hardy with 30 directions. And this was followed by probabilistic tracking using spherical deconvolution, which you heard about in the morning. So then we can again, so first thing that we can see here is that the contrasts look very different for the 2 areas. And then we can also compare this with silver staining to again understand the diffusion contrasts and their structural basis in the cortex. So you can see these silver stained images and their layer specific contrasts. So these figures basically showed or highlighted some of the examples of how we can use high angular resolution to look at microstructure in the postmortem brain. So now I'm going to move and talk a little bit about the effect of spatial resolution and its importance, especially for preclinical imaging. So these are some images from the ex-vivo mouse brain, which is about 3,000 times smaller than the human brain. And on the right, I've shown the effect of increasing spatial resolution while maintaining the same angular resolution. So here we can see that as we start to go down to about 55 microns, we start to see these very fine fibers in the striatum, which are relatively less easy to differentiate at lower resolutions. So again, why bother and why is this important? So this basically allows the diffusion contrasts in different mouse models to be directly compared with, for example, tracer injections, which are more difficult to do in the whole human brain. So here I've shown an example of using tractography in the reeler mouse cortex. So this study basically looked at thalamocortical projections using probabilistic tractography with Hardy. And on the right, these are compared with tracer injections in the same mice to understand the aberrant cortical connections in the reeler mouse. And this is basically in vivo Hardy using 30 directions. So now if we go one step further and look at the embryonic mouse brain, which is really tiny. So these are just some examples showing diffusion microimaging and what can be done in the embryonic mouse cortex. So this is these are track density images generated from data acquired at 52 micron resolution. And here we can see different developmental zones in the embryonic cortex that can be differentiated on the basis of diffusion contrasts. And on the right, I've just shown some of the technical aspects involved in the acquisition using basically grays with correction of the phase modulation that I mentioned before, and also the point split function for the resolution, which is shown on the top. And so why is this useful? So we can now compare diffusion contrasts directly with microscopy images to understand their real microstructural basis in the cortex. So here, for example, on the left, we can see the diffusion orientations that are resolved in different developmental zones. And we can see that these correspond to the orientations of radial glia and microtubules, which are specific to neurites in the developing cortex. So in the last section of this talk, I'm going to introduce diffusion using oscillating gradients and show what we can do with it. So far, we've seen the effect of changing the gradient orientations and changing the gradient strength, or B value. But what happens if we change the diffusion time? So diffusion time basically here is just the time interval between these two gradient pulses. And as I've shown in the electron microscopy image on the right, we can see that there are microscopic barriers at different spatial scales that restrict diffusion in the brain. For example, intracellular structures as well as extracellular structures. So basically, if we image at longer diffusion times, the water molecules have a higher probability of encountering more barriers or barriers over longer spatial scales. So the question is, can we get any additional information if we change the diffusion time? So this is more of a technical slide, but this is just basically showing that because of this restricted diffusion, we get a diffusion spectrum that is not flat or constant, but it actually looks like this red curve that is shown here. And the point to note in this slide is that the shape of this curve or the frequency characteristics of this curve are basically directly influenced by the tissue microenvironment. So in diffusion MRI, we usually measure at long diffusion times, which is close to zero frequency, and that's why we get really low ADC values. So sampling of the spectrum at higher frequencies can actually provide further insights into tissue microstructure. And one way to do this is to use oscillating gradients. So the idea is very simple. We basically replace these pulse gradients with oscillating or high-frequency oscillating gradients. And here I've shown these, the very familiar spin echo sequence using pulse gradients in white, and then oscillating gradients at different frequencies of 50, 100, and 150 hertz. So effectively, this just allows us to achieve shorter diffusion times. And this is a simulation study that used basically a digital phantom of packed cells. And in the plot on the right, we can basically see the predicted behavior of ADC with frequency. So basically, the ADC increases with frequency in two different tissues. And in this case, these two tissues were basically modeled to have different nuclear to cell volumes or sizes. So why is this useful? The brain, as we know, has quite a heterogeneous microstructure with several different kinds of cell populations. So can this method then actually provide any useful information about microstructure in the brain? So these are some results in the mouse hippocampus. So on the left are T2 weighted images, and on the right are ADC maps at increasing frequencies. So here we can see layer-specific enhancement in ADC, and then if we compare this with histology, we can see that these layers actually correspond to the granular and pyramidal layers in the hippocampus. And the point to note here is that these are the two most densely packed layers in the hippocampus. So basically, we end up getting layer-specific contrast with increasing frequency. And these are OGSE contrasts or oscillating gradient spin echo contrasts in the mouse cerebellum. So here again, we see that with increasing frequency, we get specific ADC enhancement in the granular cell layer as compared to the molecular layer in the cerebellum. And we can more clearly see this in the plot on the right, which shows significant enhancement with increasing frequency as compared to what we get using conventional diffusion MRI with long diffusion times. And if you look at TAPI staining at the bottom, we can again see that the cerebellar granule cell layer consists of very densely packed cells, which is in contrast with the molecular layer, which is basically acellular. So this slide basically shows frequencies-dependent contrasts in the entire mouse brain. And what is interesting here is that these contrasts seem to correspond very closely to the intensity of Nissl staining in the brain, which could suggest that they are sensitive to cellular density in brain areas. And this bar graph at the bottom basically shows the increase in ADC versus frequency for across different cellular layers in the brain. So for example, here we can see, I don't know if you can see this. So for example, here we can see enhancement specifically in the hippocampal layers, the cerebellum, and also some of the layers of the cortex that can be seen here. So this is just to show that how we can use diffusion MRI to try and get new contrasts for mapping of the brain. And then we can also use this technique to look at pathological alterations in the brain. So this is an example showing OGSE diffusion MRI in brain tumor in a mouse brain. So here, these are images at increasing frequencies from top to bottom. And here again, we can see increase in ADC specifically in the tumor region. So this can be used to differentiate the tumor area from surrounding brain tissue. And also we can see this tumor-specific enhancement of ADC in the plot on the right. And this is another example showing how we could use oscillating gradient methods to look at hippocampal neurodegeneration after injury. So these are mice after hypoxia ischemia. And on the left, I've shown maps of, or fitted maps of ADC versus frequency. And here again, we can see that there are there's a localized loss of contrast in the ipsilateral hippocampus, which we can see here and possibly here. And then if we go ahead and do histology of these same mice and compare with H&E staining, so we can see that this corresponds very well to localized regions that show cell loss after hypoxia ischemia. So this is an example to show how we can use diffusion to actually look at neurodegeneration in mouse models. And then this is an example showing a cerebellum from a monkey brain. And this again shows significant enhancement or highlighting specific to the granule cell layer, which we can see here. And this is using oscillating gradients at 4.7 T. So next, can we use OGSE DMRI to look at in vivo human brain? So a major challenge here is the very strong gradient requirements for the oscillating gradients. And one way to circumvent or to mitigate this requirement is to use trapezoidal waveforms that I've shown here. So on the right is an image from a very recent study. So this shows a specific enhancement of ADC, which we can see here in the corpus callosum of the human brain at frequencies that can be achieved on a clinical scanner, in this case at 3 Tesla. So lastly, this slide shows OGSE contrasts in layers of the human cerebellum. So here again, we can see significant enhancement specific to the granule cell layer, which is also very nicely highlighted in both of these maps here, which are basically just fitted maps of ADC versus frequency. And we can also see in this plot here that this ADC increase is actually very specific to the granule cell layer, whereas the other layers, including white matter, show only a minimal increase in ADC with frequency. So this leads to very different very different contrasts at higher frequencies compared to what we can get using conventional diffusion MRI or DTI with pulse gradients. And finally, we can also compare this with histological H&E staining in the same brain. And here we can again see the excellent correspondence between OGSE contrast and histological staining. And this also shows that this contrast is quite specific to the intensity of nucleus-specific staining in the brain. So in this lecture, I've covered some of the basics of preclinical and postmortem diffusion MRI. And try to show some of the more practical applications of a lot of techniques that we heard about this morning. And the take-home messages are, so diffusion can be sensitized to varying aspects of tissue microstructure. And you can choose acquisition and modeling strategies that basically best suit the biological question to be answered. So for example, we can pick and choose between DTI versus Hardy or even DSI if you are feeling adventurous. And then we can decide between angular or spatial resolution as well as deterministic versus probabilistic tracking. And then in addition to B-value and gradient orientation, the diffusion time can also play an important role. And diffusion MRI using oscillating gradients can provide more additional or more specific or additional information about spatial scales in tissue microstructure. And these can lead to some very interesting contrasts in the brain. So thank you for your attention, and I would like to thank OHBM and Flavio for the invitation. And if there are any questions, please feel free to email me, and the address is given here. So thank you.
Thank you very much, Manisha. Fantastic talk. Very nice. I'm sure there are questions, because if not, there are no questions. I have a question. So another question? OK. So how do you prepare the sample for the acquisition? Do you do any tissue preparation, or can you just explain briefly how we can get these pictures?
Yeah, that's actually one of the most important steps in postmortem imaging. So basically, when we typically use fixed tissues. The problem with using fresh tissue is that they tend to degrade very soon. So with fixed tissue, as I mentioned, you have much shorter T2s as well as ADCs. So we need to rehydrate them, and this is typically done by immersing the specimen in phosphate buffered saline for at least 2 to 3 days if it's a human brain sample. So that does offset some of the effects of fixation. And then in terms of imaging, we need to make sure the sample doesn't move around, so it has to be very firmly fixed in the tube that you're using for imaging.
Okay, thank you. Sorry, I'm very curious, so I have another question, if you don't mind. And about the oscillating gradient, do you have any issue about vibration, or because I said the picture looks very still, very no. So how do you compensate for vibration? Do you have any kind of holder or something? It does cause heating of the gradients, and we've also ended up burning the gradients sometimes. But so what we can do is typically also because these scans are at different frequencies, so they are a little bit longer than the conventional DTI scans. So we do see motion or displacement of images across different frequencies. This could be because of vibration or field drift or even the sample moving because the gradients are really strong. So what I typically do is to just use a simple widget registration between images across different frequencies so that when we generate a frequency-dependent map, it's all in the same spatial space. Okay, thank you very much.
Thank you.
Okay, I think we can move now to the